Now look at this.
What's up, penguins? Today we're going to do topic 2.1 on the subcellular components. So in this video, we're going to talk about the structure and function of a lot of the different organelles found inside of cells. So let's first talk about the ribosome. So the ribosome is made up of rRNA plus protein. rRNA stands for ribosomal RNA. There's two parts to it. There's a small subunit and a large subunit. The small subunit is going to bind to the mRNA, that messenger RNA, and the large subunit is going to bind to the tRNA, or your transfer RNA. mRNA is going to be transcribed from DNA, while your tRNA is going to be bound to amino acids to bring them to the ribosome for the process of translation. Ribosomes have a big function in order to synthesize proteins, as we said before, with translation. And so these free ribosomes are going to allow for us to make cytosol-like proteins, or proteins for the cytosol. The ribosomes show evolutionary significance due to the fact that all cells, prokaryotes, eukaryotes, all have ribosomes. And so that shows evolutionary significance because they've all descended from a single cell, and that single cell must have had that ribosome in it. So there's two different types of endoplasmic reticulum, or ERs we're going to call it. You have your rough ER, which is going to be studded with ribosomes. Ribosomes are going to be for protein synthesis, and so having these ribosomes attached to our rough ER is going to allow us to make membrane proteins or proteins for secretion. Our smooth ER is going to be these little tubules that come out of the nuclear envelope. Again, they're made up of membrane, and they have three main functions. Those three functions are for calcium storage, detoxification, as well as lipid synthesis. Once those membrane proteins leave our rough ER, they're going to go over to our Golgi. So the big function for our Golgi is that it's going to package and modify different products and then help to sort it to set it to its appropriate location. So for example, when we have those hydrolytic enzymes, they're going to fuse off and become lysosomes. Or if we have proteins for secretion, they're going to go to the plasma membrane to be secreted. So this is kind of where we sort as well as package and then modify any of those products. As you see, the Golgi is made up of these different little sacs. There is a cis-facing side that faces our rough ER, and then a trans-facing side that kind of points toward our plasma membrane. The mitochondria, as you also nicely call the powerhouse of the cell, which you will not use on the AP exam, is going to be the site of oxidative phosphorylation. So if you notice, there's this inner membrane called a cristae. So this is inner membrane is highly folded, and due to that increase of surface area, there's more sites for this oxidative phosphorylation that's going to occur, and so that structure leads to the function of increased amount of ATP that can be synthesized. So your big thing about mitochondria is this is the site of ATP synthesis. So apparently I'm not going to finish this in one video, so stay tuned for part 2, and remember, AP Bio Penguins are just success. Bye y'all.